Genes quietly influence how the brain interprets the world long before a conscious decision is made. When a person encounters uncertainty, challenge, or potential danger, the brain must rapidly decide whether to approach or avoid. This decision is not neutral. It is shaped by inherited differences in how neural circuits respond to reward and fear. For some individuals, uncertainty activates systems associated with curiosity and anticipation. For others, the same uncertainty triggers caution and stress. These responses feel like personality traits, but they are rooted in biology. At the center of this process is the balance between reward circuitry and threat detection. The reward system, driven largely by dopamine pathways, evaluates potential upside. When this system is highly responsive, the brain assigns greater value to possible gains. Risk begins to feel energizing, even necessary. Taking a chance produces a subtle sense of forward motion, a pull toward action. In contrast, the threat system, heavily linked to the amygdala and stress hormones like cortisol, evaluates potential loss. If this system is more sensitive, the brain prioritizes safety and predictability. Risk feels heavy, costly, and mentally draining. Genes help determine how easily each of these systems activates. Variations in genes related to dopamine receptors can increase or reduce how strongly reward is felt. A stronger reward signal doesn't mean reckless behavior. It means the brain learns faster from potential gains than from potential losses. Meanwhile, genetic differences affecting stress reactivity influence how quickly fear responses arise and how long they last. A highly reactive stress system may interpret ambiguous situations as dangerous, even when objective risk is low. What's important is that these processes happen beneath conscious awareness. People often explain their choices using logic or values, but the emotional weight assigned to an option has already been biased by neural activity. Two people can assess the same opportunity with the same information and arrive at opposite conclusions, not because one is smarter or more disciplined, but because their brains assign different emotional costs and rewards to the same outcome. This biological bias also shapes learning. When risk leads to success, a brain that strongly encodes reward will reinforce future risk-taking. When risk leads to failure, a brain with heightened threat sensitivity will strongly encode avoidance. Over time, these patterns compound, creating stable behavioral tendencies that feel like identity. Someone begins to think of themselves as a risk-taker or a cautious person, without realizing that these labels emerged from repeated neural reinforcement. However, bias is not compulsion. Genetic influence operates more like a volume knob than an on-off switch. It increases the intensity of certain signals, but does not eliminate choice. The prefrontal cortex, responsible for planning and impulse control, can override emotional drives when properly trained. Experience, reflection, and deliberate exposure can reshape how reward and fear are interpreted. A person genetically inclined toward caution can gradually reduce threat sensitivity through controlled challenges. A person drawn to risk can learn to pause and evaluate long-term consequences. Understanding this framework removes moral judgment from behavior. Risk-taking is not inherently brave, and caution is not inherently weak. They are adaptive responses tuned differently across individuals. In environments that reward exploration, high reward sensitivity can be an advantage. In environments where stability matters, strong threat detection can be protective. Problems arise only when a biological bias operates outside the context where it is useful. When people recognize that their instincts are shaped by inherited neural tendencies, they gain leverage. Instead of fighting themselves or labeling their reactions as flaws, they can design systems that account for their biology. Decision-making improves not by suppressing instinct, but by understanding where it comes from and how it influences perception in real time. Dopamine plays a central role in how motivation is generated and sustained, but its function is often misunderstood. It is not simply a pleasure chemical. Instead, dopamine is about drive, anticipation, and the willingness to move towards something uncertain. Genes that regulate dopamine production, release, and receptor sensitivity shape how strongly a person feels motivated by potential rewards, especially when outcomes are not guaranteed. Some individuals inherit dopamine systems that respond strongly to novelty. New experiences, unfamiliar environments, and uncertain outcomes trigger a surge of motivation. The brain interprets novelty as opportunity. This doesn't mean the reward is guaranteed, but the anticipation itself feels compelling. For these individuals, routine can feel mentally draining, while change feels stimulating. Risk, in this context, becomes a pathway to engagement rather than a threat to stability. Other people have dopamine systems that are less responsive to novelty and more responsive to predictability. Their brains release dopamine more reliably when outcomes are clear and structured. For them, motivation comes from mastery, consistency, and known rewards. Uncertainty does not provide the same motivational lift. Instead, it may feel like wasted effort or unnecessary strain. These differences are subtle, but over time, they shape major life patterns. Dopamine-related genes also influence how quickly motivation rises and falls. Some nervous systems generate fast, intense bursts of drive that fade quickly. This can create a pattern of enthusiasm followed by restlessness once novelty wears off. Risk-taking, in this case, is not about danger, but about reactivating motivation. The brain seeks stimulation to re-engage dopamine circuits. In contrast, slower, more sustained dopamine signaling supports long-term effort and delayed gratification, reducing the pull toward impulsive risk. Importantly, dopamine interacts with learning. When a risk leads to a positive outcome, dopamine strengthens the neural pathways associated with that behavior. The brain essentially tags the action as worth repeating. If dopamine signaling is strong during anticipation, even small wins can reinforce future risk-taking. Over time, this creates a feedback loop where uncertainty itself becomes motivating, regardless of outcome magnitude. This mechanism explains why some people are drawn to entrepreneurial ventures, competitive environments, or high-variability careers. The uncertainty inherent in these paths continuously stimulates dopamine systems tuned for novelty. The same logic applies to creative pursuits, exploration, and innovation. Risk is not experienced as recklessness, but as alignment with how the brain stays engaged. On the other hand, when dopamine signaling is less sensitive to novelty, risk does not produce the same learning signal. Even successful outcomes may not feel sufficiently rewarding to justify the effort. These individuals are not unmotivated. Their motivation is simply optimized for different conditions. They thrive in environments where effort reliably leads to outcome and where progress can be measured incrementally. Dopamine genes do not act in isolation. They interact with stress systems, social feedback, and past experiences. A person genetically inclined toward novelty seeking may become more cautious after repeated negative outcomes. Conversely, someone naturally risk-averse may increase novelty seeking if rewarded consistently in a supportive environment. Genetics sets the sensitivity, not the script. What often gets mislabeled as impulsivity or ambition is, at its core, a difference in how motivation is generated. Some brains need uncertainty to come alive. Others need clarity. When people misunderstand this, they try to force themselves into motivational strategies that don't work for their biology. The result is frustration, burnout, or unnecessary self-criticism. Recognizing the role of dopamine-related genes reframes risk-taking as a motivational style rather than a character flaw. It explains why advice that works for one person fails for another. Telling a novelty-driven brain to just be consistent can feel suffocating. Telling a stability-driven brain to take more risks can feel overwhelming. Motivation is not about willpower alone. It is about aligning behavior with how the brain is wired to seek reward under uncertainty. Moral sensitivity often feels like a product of values, culture, or conscious reasoning. But beneath those layers lies biology. Genes influence how strongly the brain responds to social cues such as fairness, suffering, trust, and cooperation. These responses form the emotional backbone of moral behavior. Before a person reasons about what is right or wrong, their nervous system has already generated signals that guide attention, discomfort, or concern. Key to this process are genes that regulate neurotransmitters and hormones involved in social bonding and emotional regulation, particularly oxytocin and serotonin. Oxytocin plays a major role in empathy, attachment, and the ability to perceive others as emotionally significant. When oxytocin signaling is strong, the brain more readily picks up on facial expressions, tone of voice, and subtle signs of distress. Harm to others is not abstract. It feels immediate and personal. Moral violations register as emotional disturbances, not just intellectual disagreements. Serotonin, on the other hand, helps regulate mood, impulse control, and sensitivity to social norms. Genetic variations that affect serotonin availability can influence how deeply a person feels guilt, shame, or responsibility. For individuals with heightened serotonin-related sensitivity, breaking a moral rule produces internal discomfort that can be as powerful as physical pain. This discomfort acts as a braking system, discouraging actions that might disrupt social harmony. These biological systems shape how morality is experienced, not which moral framework is adopted. Two people may both believe in honesty, but one experiences lying as emotionally distressing, while the other evaluates it more cognitively. The belief is shared, the internal experience is not. This difference often leads to misunderstanding, where one person assumes the other lacks morals, when in reality they simply process moral information through a different neural lens. Empathy itself varies in intensity due to genetic factors. Some nervous systems naturally simulate the emotional states of others with high fidelity. Observing someone in pain activates overlapping neural circuits, creating a shared emotional state. This makes compassion almost reflexive. In contrast, other brains require deliberate attention and reasoning to generate the same response. Empathy in this case is not absent, but it is less automatic. These differences influence moral priorities. High empathy sensitivity often correlates with strong concern for harm reduction and fairness. Moral decisions are guided by how outcomes affect others emotionally. Lower empathy sensitivity does not imply indifference. It often leads to a greater reliance on rules, logic, or principles rather than emotional cues. Such individuals may prioritize consistency, justice, or long-term consequences over immediate emotional impact. Genes also affect how people respond to moral conflict. When faced with competing values, some brains experience intense internal tension. The emotional weight of the decision can feel overwhelming, driving a strong desire to resolve the conflict quickly. Other brains tolerate ambiguity more easily, allowing moral dilemmas to be approached analytically without significant emotional distress. This shapes not only decisions, but communication styles during ethical disagreements. Importantly, moral sensitivity is context-dependent. Genetic predispositions interact with upbringing, culture, and repeated experiences. A supportive environment can amplify empathy-related tendencies, while chronic stress can blunt them. Likewise, reflection and practice can strengthen moral awareness even in individuals whose biology does not naturally emphasize emotional resonance. Understanding the biological roots of moral sensitivity changes how moral differences are interpreted. Disagreement is no longer seen solely as a failure of character or values, but as a difference in emotional signal strength. Some people feel morality loudly, others feel it quietly. Both can act ethically, but they arrive there through different internal routes. When people recognize that their moral reactions are shaped by neural sensitivity rather than pure choice, judgment softens. Instead of assuming others are careless or overly sensitive, they can appreciate that moral experience exists on a spectrum. This awareness allows for more productive dialogue, better self-understanding, and a clearer separation between values and the biological intensity with which those values are felt. Moral disagreement is often misunderstood as a clash of values, when in many cases it is a difference in emotional intensity rather than belief. Two people can agree, in principle, on what is right or wrong, yet experience moral situations in dramatically different ways. This happens because genes influence how strongly moral signals are felt in the brain. The value may be the same, but the internal volume is different. When a moral issue arises, the brain does not process it as a single event. Emotional circuits activate alongside cognitive ones. For some individuals, emotional systems respond rapidly and forcefully. Violations of fairness, honesty, or care trigger immediate discomfort, anger, or distress. These reactions feel automatic and undeniable. The person is not choosing to feel strongly. Their nervous system is simply generating a powerful signal. For others, the same situation produces a much quieter emotional response. The moral issue is still recognized, but it does not dominate attention. Instead, cognitive systems in the prefrontal cortex take the lead, weighing context, consequences, and trade-offs. This does not mean the person cares less. It means the emotional charge attached to the value is lower, allowing more mental space for analysis. This difference in emotional volume is largely shaped by genetic variations affecting neurotransmitters and neural connectivity. Systems involved in empathy, threat detection, and social bonding can be more or less reactive. When these systems are highly sensitive, moral violations feel urgent and personal. When they are less sensitive, moral decisions feel more abstract and flexible. Neither mode is inherently superior. Each has strengths and limitations. Problems arise when people assume that emotional intensity equals moral depth. A person who reacts strongly may believe that their feelings prove moral clarity, while interpreting a calmer response as indifference or lack of principle. Conversely, a person who approaches morality analytically may view intense emotional reactions as irrational or exaggerated. Both interpretations miss the underlying biology driving the experience. These differences become especially visible in group discussions, social conflicts, and ethical debates. One person may feel compelled to act immediately, driven by emotional discomfort. Another may want time to reflect, gather information, and consider alternatives. When emotional intensity is mistaken for urgency, patience can be interpreted as apathy. When restraint is mistaken for coldness, trust erodes. The same dynamic appears in personal relationships. A moral boundary that feels non-negotiable to one partner may feel situational to the other, even if both agree on the principle behind it. The conflict is not about values, but about how strongly the nervous system reacts when the boundary is approached or crossed. Without this understanding, both sides may feel misunderstood or morally judged. Emotional intensity also affects memory and learning. Strong moral emotions imprint experiences deeply, making certain violations unforgettable. This reinforces future reactions, creating a feedback loop where similar situations trigger even stronger responses. Lower emotional intensity leads to more flexible updating of moral judgments based on new information. Again, this is not about right or wrong, but about how the brain prioritizes information. Recognizing that moral experience operates on a spectrum allows for more accurate self-assessment. A person who feels moral outrage quickly can learn to pause before acting, understanding that the intensity is a signal, not a command. A person who feels less emotional urgency can learn to check whether analysis is unintentionally overriding empathy. Both adjustments improve ethical decision-making. When emotional volume is separated from moral value, conversations change. Disagreement becomes less personal and more informative. People can acknowledge shared principles while respecting different internal experiences. This reduces defensiveness and opens space for cooperation. Morality becomes not a contest of who feels more, but a process of aligning actions with values, while accounting for the biological differences that shape how those values are felt. Genes influence behavior by shaping tendencies, not by issuing commands. They create a range within which behavior is more likely, but they do not lock a person into a fixed outcome. This distinction is critical. When genes are misunderstood as destiny, people either excuse their behavior or feel trapped by it. In reality, genetic influence works through probabilities, increasing the likelihood of certain responses while leaving room for change through experience and environment. From birth, genetic differences affect how the brain processes reward, stress, emotion, and learning. These early biases make some behaviors easier and others more effortful. A person may naturally gravitate toward caution, novelty, empathy, or emotional restraint. Over time, repeated choices that align with these tendencies strengthen the associated neural pathways. What feels like fate is often the result of reinforcement layered on top of biology. Environment plays a powerful role in shaping how genetic tendencies are expressed. Supportive environments can soften biological extremes, while chaotic or stressful ones can amplify them. For example, a person with a genetic sensitivity to stress may become highly anxious in an unpredictable setting, but function calmly and effectively in a structured environment. The gene does not change. The context determines how strongly it shows up in behavior. Habits act as the bridge between biology and choice. Repeated actions reshape the brain through neuroplasticity, strengthening some circuits and weakening others. A person genetically inclined toward impulsivity can train impulse control through routines that reduce friction and increase pause. A person inclined toward emotional avoidance can build tolerance for discomfort through gradual exposure. These changes do not erase genetic bias, but they can significantly redirect behavior. Importantly, the brain learns from what is repeatedly practiced, not from what is intended. Genetics may set the initial direction, but habits determine the trajectory. Over months and years, consistent behavior can override early predispositions to a surprising degree. This is why people can change dramatically across different life stages, despite having the same genetic makeup. Culture and social norms also interact with genetic tendencies. Societies reward certain traits and discourage others. A person whose biology aligns with cultural expectations may feel naturally good at life, while someone misaligned may feel flawed. But when context shifts, those same traits can become advantages. What looks like destiny in one environment can look like flexibility in another. Awareness is the critical leverage point. When people recognize their biological tendencies, they stop personalizing them as character flaws. Instead of asking why change feels hard, they understand which systems require more support. This allows for strategic design of environments and routines that compensate for bias rather than fighting it directly. Choice enters not at the level of instinct, but at the level of structure. People cannot always control their initial reaction, but they can control what happens next. They can choose how much exposure they allow, which habits they reinforce, and which environments they place themselves in. Over time, these choices reshape behavior in ways that genes alone cannot predict. This perspective replaces helplessness with responsibility. Genes explain why certain paths feel natural, not why alternatives are impossible. When behavior is seen as a dynamic interaction between biology and experience, change becomes a process rather than a contradiction. The future is not written in DNA. It is negotiated daily through environment, repetition, and awareness. Self-awareness of genetic tendencies is one of the most powerful tools for shaping behavior. Understanding that your instincts, emotional reactions, and motivational drives are influenced by inherited neural patterns allows you to approach life with clarity instead of confusion or self-judgment. When people operate without this awareness, they often misinterpret their own behavior, labeling natural inclinations as flaws or inconsistencies. Recognizing the biological underpinnings of behavior reframes self-perception from criticism to understanding, creating a foundation for deliberate action. Genes bias the brain's responses in predictable ways, but they do not dictate absolute outcomes. Some individuals are naturally drawn to novelty, others to predictability. Some are more sensitive to social cues and moral signals, while others rely on analytical reasoning. These tendencies influence decision-making, risk-taking, and moral judgment. Without awareness, a person may constantly battle against their own instincts, trying to act opposite to what feels natural, which often results in frustration, fatigue, and inconsistent behavior. Understanding these tendencies allows a person to work with their biology rather than against it, designing strategies that leverage natural inclinations. Self-awareness also enhances decision-making by highlighting the lenses through which one experiences the world. If you know that your dopamine system responds strongly to novelty, you can anticipate moments when risk-taking may feel more compelling than the situation objectively warrants. Recognizing this allows you to pause, evaluate the potential consequences, and avoid impulsive decisions that might later be regretted. Similarly, understanding that moral sensitivity may vary genetically helps explain why some dilemmas provoke intense emotional reactions while others feel abstract. By separating the intensity of the feeling from the actual decision, you can make more balanced and consistent ethical choices. Moreover, awareness fosters more precise goal setting. People who understand their motivational wiring can align their environment and habits with their neural tendencies. For example, a person naturally drawn to uncertainty may structure their life to include challenges, creative projects, or new experiences, which keeps them engaged and productive. A person who prefers stability can design routines, clear structures, and incremental goals to maintain motivation without relying on constant external stimulation. When behavior is aligned with genetic tendencies, effort is more sustainable and outcomes are more predictable. This insight also improves interpersonal relationships. Recognizing that others may experience situations differently due to their biological wiring reduces conflict and misinterpretation. Someone whose nervous system reacts strongly to social cues may assume others feel as intensely as they do, leading to overestimations of emotional impact. Conversely, those with less reactive emotional circuits may misinterpret heightened responses as overreaction or insensitivity. Understanding these differences allows for empathy, patience, and more effective communication because behavior is seen as a product of wiring plus context rather than deliberate intention or character judgment. Awareness creates opportunities for intentional training and adaptation. Neural circuits are plastic, meaning they can be reshaped over time through consistent practice, reflection, and environmental design. Knowing your predispositions allows you to target the areas most in need of adjustment. For instance, someone naturally inclined toward risk can develop strategies to manage impulsivity, such as planning ahead or introducing structured decision points. Someone with high sensitivity to stress or social evaluation can learn techniques to regulate physiological arousal, improving performance and emotional resilience. The key is that interventions are more effective when they are personalized to the underlying biology. Ultimately, self-awareness transforms instinct from an uncontrollable force into actionable information. It allows you to recognize when a gut reaction is biologically driven, predict how certain situations will affect you, and design responses that maximize alignment with your long-term goals. Instead of being a passive recipient of genetic influence, you become an active participant, shaping how predispositions express themselves. By understanding your biological tendencies, you gain clarity, control, and flexibility, making it possible to navigate risk, motivation, and morality with intentionality rather than default reactivity. By integrating this understanding into daily life, you create a framework where biology informs strategy rather than limits potential. Self-awareness becomes the bridge between innate tendencies and deliberate action, allowing you to act with insight, align behavior with values, and navigate challenges with greater confidence and consistency. It transforms genetic influence from a source of constraint into a guide for smarter choices, sustainable habits, and more authentic decision-making.